for today's event, Dr. Rahul Suryavanshi is a research scientist at Gladstone University, San Francisco, California, United States. President ISC, Dr. S.H. Bagade. Convener of today's event, Dr. Mrs. S.S. Khandare, Head Department of Microbiology. All head of the departments, respected faculty members of different institutes, and all dear participants. A very good morning to all of you. First, let me introduce myself. I am Dr. Bhavna Mohite, Assistant Professor at Department of Microbiology at Bajaj College of Science, Wardha. I am very glad to welcome you all on behalf of Shiksha Mandal's Bajaj College of Science for this motivational talk of Successful Innovator, which is jointly organized by Department of Microbiology and Institution Innovation Council. Foremost for the speaker, I will briefly introduce our parent organization, Shiksha Mandal. Shiksha Mandal is a century-old Bajaj educational trust. It is well known for providing the quality education steeped in nationalistic ethos and human values at affordable cost. Bajaj College of Science is one of the colleges run by Shiksha Mandal. It is the granted aid institution established in 1962 with the prime objective of catering science education at Wardha. It is the linguistic minority and autonomous college affiliated to Rashtrasanta Tukaroji Maharaj, Nagpur University, and offers undergraduate, postgraduate, and PhD program in science. The college has been awarded with the status of first institute accredited by NAC in entire Vidarbha. College with potential for excellence by University Grants Commission, Star College by Department of Biotechnology, re-accredited A grade with a CGPA of 3.21 by NAC. It is the Rusa Beneficiary Institute and selected by MHRD as Institution Innovation Cell, and many more. So this is in brief about the host institute for your information, sir. We appreciate you all for joining us for today's event. Ministry of Education, MOE, Government of India has established this MOE Innovation Cell to systematically foster the culture of innovation among us all higher education institutes. The aim of this program is to encourage, inspire, and nurture the young minds for translating their ideas into innovation. With this change, we organize today's event, and I hope this talk will bring a positive transformation in the students, their approach towards their ideas, research projects, and groom them future ready and out of box thinkers. Today's program today's program is a small step to motivate the young minds to come up as student innovators. I request convener of the program, Dr. Mrs. S.S. Khandare, Head of Microbiology Department, to briefly give the idea behind today's program and to give her introductory remarks. Over to you, madam.
Thank you, Dr. Mohite. Am I audible, Dr. Mohite?
Yes, madam. Yes.
Okay, thank you. Thank you very much. Very good morning to one and all present over here.
You are not audible, Mrs. Khandare and Bhavna, both of you is partly audible and partly not. So,
okay, sir. So we'll try to improve the audibility.
Hello, am I audible? Hello?
Yes, ma'am, you are audible.
Okay, thank you. A very good morning to one and all. Honorable Principal Dr. Om Mahodaya. Today's guest speaker, Dr. Rahul Suryavanshi. President, Institutions Innovation Council, IIC, Dr. Bagade. Dr. Mohabansi, Convener of IIC, the faculty members, and dear students. I am very glad that Department of Microbiology, in collaboration with Institutions Innovation Council, that is IIC, organizing the motivational talk on how we can be a successful innovator. We feel privileged as we have Dr. Rahul Suryavanshi, research scientist at Gladstone University, San Francisco, California, United States, as a speaker to talk with you, dear students. I welcome you, sir, in the Department of Microbiology, Bajaj College of Science, Wardha. Today, everybody is talking about startups and entrepreneurship. Teachers and mentors keep on telling the students regarding innovations and entrepreneurships. If you want to be a successful entrepreneur, you should be a good innovator. Our subject microbiology has got tremendous potential to go for bio entrepreneurships. What happens with you, dear students? You people only read the syllabus as far as examination is concerned. The study superficially just to get the marks in the examination. But study of the subject is a continuous process. Read not only related with the syllabus, but the subject as such in detail. To get the insights, students should think critically about the subject. Then sometimes an idea may click. What can be done? So think over and act accordingly. All the work done at laboratory level should reach to the end user. So there should be a link in between, and this link may be a entrepreneur. Now today's talk is specially arranged for you, a motivational talk. Dr. Suryavanshi is a well-known researcher. He has done a lot of research in different areas, and one of the area among his work is on water-soluble pigment prodigosin. From the bacterium Serratia marcescens, this product may find its way in commercial use in sunscreens or as nematicidal or as a mosquito larvicidal or pupacidal. This is a wonderful work. Especially in such a period where the world is suffering from deadly disease like malaria and trying to search a new remedies to overcome this deadly disease. Today, there is a need to go for biological way to kill mosquitoes, as there are various deleterious side effects of chemical insecticides. Earlier people also they tried for endotoxin of Bacillus thuringiensis to control mosquitoes at the larval stage. One of our PG students also work on prodigiosin production from bacteria Serratia marcescens and its commercial use with a very good publication. So once again, I welcome Dr. Rahul Suryavanshi in Department of Microbiology, Bajaj College of Science, Wardha. And I would like to stop here. Thank you. Thank you very much. Over to you, Dr. Mohita.
Thank you, madam. Just a little housekeeping before we get started. So it's instructions for all the participants. So please keep your audio and video on off mode, so it will not distract the speaker. And secondly, please free to submit your questions at any time simply by writing in the chat box or in the comment section of YouTube live streaming. We will be monitoring that questions and we'll try to take the maximum questions. I will collect that and address them to the presenter at the end of the session. So, and the speaker will be really happy to respond all of you. Today's program is possible only because of the continuous encouragement and guidance of our principal and who is the president of today's event, Dr. Om Mahoday sir. I request Principal Mahoday sir for the presidential address. Please, sir, over to you. Are you getting Mahoday sir? I request Principal Mahoday sir for his remarks.
All of you are non-audible. Audio quality is very, very poor. I don't know why, how can the students would get it. Am I audible to all of you?
Yes, sir.
Very good morning to all of you. Mr. Rahul, Dr. Rahul, research fellow who has been associated with us. I welcome him.
Thank you, sir.
In fact, today's program is one of the series program under the IIC, Institutional and Innovation Council, Government of India. In fact, such activities which has been promoted are needed to energize energize or catalyze the research activities in the various field or in the academic scenario or academic platform. As a matter of fact, whenever we needed an improvement in any of the system, education is one of the aspects in it. We have to peep into it. We have to search something new. And we have to modify the things. We have to see the alternatives. And that's exactly what is called the innovation and research. In our country, the ecosystem which has been around the different systems, we are talking about the educational system, needs much impetus. We have to give the much impetus on the research activities. And the precursor of research is 100% is the innovation process. And innovation start coming when you start imagining the things in your mind. The students are sitting with this, are participating in this. The imaginations may be converted into the innovations, and the innovation then proceed into the research activity and ultimately the new finding. So the basic thing is that you should be in the thinking on why, how the imagination comes in mind. Whenever you see the sequence, for example, Dr. Rahul is with us. He would be sharing his experiences and his work with us. It will definitely give a trigger to the young minds that how to go, how to think, and how to proceed for the innovation. In fact, in our country, there are, if I talk of, there are challenges, societal challenges are there. For example, education we are talking about, cleanliness, clean water. We didn't have the clean water in our country at all the places. Moreover, the sanitation, healthcare, even air quality, energy, transportation. These are the number of things which and infrastructure, various infrastructures, which are in fact needed to be improved in our country. And in education, whether it is a school level or higher level education, the innovational ecosystem is badly needed. In fact, the government has started the process triggering these issues, and therefore this IIC is formed. So this is a very good activity. It is run by the HRD and adapted by the different colleges to conduct these programs in their colleges, in their institutions. And we are organizing, I think it's most probably it is the third or fourth one IIC program. So I hope that today would be the center of discussion. We have the microbiological aspect in innovation. Dr. Rahul would be speaking around these issues and would throw a light. As I've told you, the students, those who are listening today, would definitely be getting motivated and would be having certain areas where they can have their innovations. These are the such activities are there to improve the ecosystem of the colleges, universities, as far as the education is concerned. So with these words, I again welcome Dr. Rahul here for this good program. The topic is a very wonderful topic, which is in need of an hour. All the participants. I expect them that they would be very serious about this issue, and they would discuss it, and definitely they will raise their questions, what they have got it in their mind. They can directly, because I am saying that Rahul is very young man, is very enthusiastic, looking to be very enthusiastic, like you, dear students. So you must ask the different questions to him, if at all if you're not in difficulty, and get it clearly. With these words, I congratulate to Department of Microbiology and the IIC officials for organizing this program and give me this opportunity to talk. Thank you very much. Over to Bhav.
Thank you so much, sir, for the encouraging words. Sir has nicely put what is the need of innovations and what the area to be focused for the innovation. And certainly, sir, we hope that Dr. Rahul's talk will motivate the students and certainly they give some part to their ideas. Thank you so much, sir. Today, our resource person is Dr. Rahul Kushal Suryavanshi. To highlight the creditability and expertise of the speaker on the subject matter, I briefly introduce him. It is indeed a pleasure to introduce Rahul. I feel very proud because I personally know Dr. Rahul, particularly as he was my colleague and we worked together under the guidance of our mentor, Dr. Satish Patel at North Maharashtra University. So it's really a nice experience to work with Rahul. I saw him working with passion and hard work and going as researcher. Dr. Rahul is presently working as research scientist at Gladstone Virology Institute at San Francisco, USA. He is presently working on pathogenesis of SARS and COVID virus in mice and Syrian hamsters. He has past experience as postdoctoral associate at University of Illinois at Chicago. He was selected as Erasmus Mundus Fellow at University of Perpignan, France. Rahul was awarded with Junior and Senior BSR Fellowship of UGC at North Maharashtra University. He worked as laboratory technician at National AIDS Research Institute of ICMR at Pune and as research associate at Symbiosis Bioware, which is now recognized as Lonza Biotech. Dr. Rahul got the research funding such as ISPB grant, SERB National Postdoctoral Fellowship. He has 55 research publications on his credit in renowned journals, to mention a few, and it is actually a proud to say like that Rahul has published various his research papers in journals like Nature, Communication, Journal of Virology, Pathogens, Natural Product Research, and Bioorganic Chemistry. With his some of the rewards or honors I want to mention here, particularly Gladstone Institute of Virology Award of Excellence for the year 2021 has been announced to him recently, and in the month of January, he will be awarded with that. He has been awarded with Kretschmann Postdoctoral Research Award at Ben Gurion University of Negev, Israel. He was selected for the Erasmus Mundus Emeritus Fellowship by the European Union. He got the International Travel Grant by ICMR and completed the Biotech Industrial Training by Biotech Consortium India Limited, Government of India. So this is a lot and many more things, but I think it's a nice brief introduction of Dr. Rahul. After this brief introduction, we move on further, and without any further ado, we will turn the time over to the presentation. Let's get started with the presentation by Dr. Rahul Suryavanshi, for which we all are eagerly waiting. I request Dr. Rahul to please share the screen and proceed for the presentation. Please, Dr. Rahul.
First of all, can you hear me clearly?
Yes, Rahul, your voice is clear.
Oh, yeah. Thank you very much, Bhavna ma'am, and all the honorable people from the organization.
I'm sorry.
I'm feeling very sorry.
Yes, sir.
Am I audible?
Yes, yes, Dr. Rahul.
Okay. Yeah. I'm feeling very privileged to be here actually to talk to my friends who I would like to motivate them to come into the science because it is need of an hour, as we can see the pandemic, recent pandemic, which is still ongoing, and only researchers can deal with it to save the millions of lives. So I would proceed with my presentation. First of all, what is motivation? One more thing I want to make it clear. I have never talked on motivation before, but I think uh, there is some background noise. May I request all of you to mute yourself so that I can concentrate on the presentation, please. What is motivation? Motivation is the process that initiates, guides, and maintains the goal oriented behavior of anyone for doing the thing they are aiming for. The fact about motivation is also that a motivation is a fire from within. If somebody else is trying to motivate you, then he can motivate you only briefly. Like you can feel the motivation for a very short period of time. But if you want to keep yourself motivated, the fire should begin from yourself. And for that purpose, we need to find the motivation um, in our day-to-day activities, in our day-to-day life, from millions of things that we observe around us. Each has a potential to motivate us. And I'm going to share my story um, of like from where I got motivated to do different things uh, or to achieve my path of getting here and to do science. So starting with who am I? So I am a research scientist based in San Francisco, California, and I'm working in a very resourceful working environment in University of California, San Francisco. And my institute, Gladstone Institute, is affiliated to UCSF. And within a Gladstone Institute, I'm in the Department of Virology. And within department, I'm working under our director, Melanie Ott, who is in charge of our lab. We are a big lab with almost 22 researchers, including graduate students, postdocs, and research scientists. So as you can see here, we work on COVID-19, so we need to wear papers. If you would like to visit, you can visit our lab website, and there you can get details about like what all topics we work on. It's not only coronaviruses, but also HIV, Zika, and all other infectious deadly viruses. Uh, so what do I do? Um, I have been recruited to work on the agent that is causing pandemic currently all over the world, and we can see uh, uh, how this tiny living thing is shaking all the world and impacting the lives of almost every human being. So I work on SARS-CoV-2 and majorly discover or reveal the secrets of its high transmission and high replication. Um, the way I do it is by working with the cells, human cells, which are isolated from lungs of human origin, and uh, infecting them with the virus and checking the changes that are happening in the cells, and then translating that same research in the animals. So as you can see, and all you know, we are seeing different variants that are coming up uh, for this SARS-CoV-2, as I can enlist alpha, beta, gamma, delta, epsilon, lambda, omicron. All these variants are variants of concern. So what is variants of concern is so these are the variants, or these are the viruses that can cause significant harm to humans. And we are investigating how the pathogenesis of these viruses happens in animals and in cell culture, and if we can block their replication or entry or co-transmission by any knowledge that we can gain from these studies. So of course, this is a very interesting study, but I would like to tell you, so you already know that I work at Gladstone Institution. I wanted to let you know why there is a difference in the other institutions, or more clearly, the institutions in India. I mean, there are many institutions in India which are very ahead in their research areas, but still the scientific progress is more significant in these countries compared to our country. And I would like to highlight some differences, why that happens, and that will also motivate us where we should excel ourselves to get or to do the meaningful science. So the first thing is, in this particular area of San Francisco, prime institutions like University of San Francisco, uh, University of California, San Francisco, University of Berkeley. One second. I have the pointer. University of Berkeley, then UCSF Medical Center, and Stanford University. All these big institutions are very close by. That means like they are within a few kilometers of a distance. So whenever a research needs to be done on like a very burning topic, like for now it is SARS-CoV-2, uh, these all institutions get united together. The scientists over there, they collaborate with each other on important topics and on the topics of their mutual interest, and they do their research in a big collaboration, which actually increases the speed of their research, and they get very quick answers for the complex problems. And that's what I'm seeing here. Since the pandemic, all these research collaborations have been accelerated, and everyone is like uh, running very hard to know the answers so that it can save the mankind. The other reason for the successful science over here is the funding. uh, US contributes a very high amount of funding towards science, and that's why they have like all the patents, all the intellectual properties, and the best medical treatments in their country, um, possible because of the funding that they get. And the more they invest in the research, the more they get through the research, because their products are sold all around the world, and from there they get royalty, and they get more money. That's how they become more and more richer, and they can give quality life to their citizens. Not only about the biotechnology or biology aspect, but even the tech parks are in San Francisco. So all the headquarters of big technical companies like Google, Facebook, Apple, they are all here. and that makes this area more resourceful. They gather all the talent from all over the world, different parts of the world, and they bring them here uh, to get their work done more smartly. You can also see this company called Gilead. This is a biotechnology company. Basically, this company have all the scientists. Basically, it is a pharma company, but they invest in science more. And the product of this company is remdesivir, which have saved a good um, amount of lives throughout the world during this pandemic. I mean, remdesivir was able to control the virus replication very partially, but it was good enough to um, decrease the rate of hospitalization for people across the world. So likewise, in case of technology, in case of science, in case of collaborations in between the scientific institutions, and this area is well ahead, and that's why they are better in science. So what's the output? As you can see, I mean, I haven't listed all the Nobel laureates from this area. These are only very few, but as you can see, there are many Nobel laureates that are from this area. Almost every year, um, this area gets the Nobel Prize. Uh, I would like to highlight Dr. Shinya and Dr. Jennifer Dardena, who are actually scientists from the same institution that I am, and we are doing work in collaboration with Jennifer Dardena, uh, who has investigated the CRISPR-Cas9 system, which have revolutionized the research across the world. It has benefited many medical therapies and whatnot. And we are currently working with her on the Omicron variant that is popping up in the area. So as you can see, this is the research output by the area, and all these things are possible uh, anywhere in the world, including our country. Um, but of course, we need to fulfill all the requirements that are essential to do that. I'm sorry, I will just adjust the light because it's motion based. So I need to move to put on the light. I will come back. As you can see, this is a drawback of more technology driven institution. So the thing is, the question is, how did I reach here? It is a exciting journey for me, of course. A motivation is such a subtle thing which can be or should be different for each and every one of us. But I will try to highlight my motivation, of like how I reach here, what obstacles I faced, and how I meet those obstacles in the past 12 to 13 years. So the journey so far, my journey actually started from North Maharashtra University, Department of Life Sciences. Then I went to France for doing postdoc, then to University of Illinois at Chicago, again for doing the postdoc, and then I'm here now, joined as a research scientist at UCSF San Francisco. So how it started, I'll explain from the beginning. So I would like to specially highlight that I was not a very intelligent student in my college days. Mm, You will be, uh, most of you will be happy to know that I secured only 53.67% of marks in my 12th, same in the 11th standard as well. So of course I was not intelligent by then. Then I also invested 1 year for preparing my medical for the medical entrance test, which of course I failed. Uh, then there was like um no choice, and I had to do bachelor's in science. But indeed, that decision was very fruitful, that you will see shortly. Bachelor in science, because I had affection towards biology. I selected the subject called biotechnology. I had all this attraction toward DNA, RNA, and molecular biology, basically. So I got admission to DNCVP College in Jalgaon, and then um, the interesting fact about the bachelor's degree is because I was not like very scholar student, but I never failed. But for the first time in my second year of bachelor's program, uh, I got 80 KT. That means allowed to keep terms, and that was like big pain for me because though I was average student, I never failed. But this is the first time when I realized that things are not right. I need to wake up. How can I get fail? But of course, university allowed me to through ATKT to get to the next year admission. But after that, I didn't look back. And in the third year of bachelor's, I did significantly well with securing first class marks. Then I took admission to masters in biotechnology, and um, here I changed style of studying actually. The things I faced or the failure I faced in my bachelor's program was due to insufficient study of not paying attention to the subject matter in detail. But when I was doing masters, paid special attention to um, reading the chapters before my teachers will teach me. Uh, Especially molecular biology, the subject that I always like to read. Uh, Molecular biology was considered to be the tough subject among my friends, but I didn't know that. In the vacation itself, I started preparing for it, and when I realized that others find it difficult, but when I read it first and then my teachers teach me, uh, I understand it better. That make me to have a habit of reading the topics beforehand the teachers teach me. And of course, because of this good habit, uh, I was able to get good score in my masters. But of course, this was not enough. After masters, there was a question like, where should I go? uh, because of having high affection towards biology, I wanted to become a researcher, wanted to do science, but there was not a proper guidance, I will say, at my level. Um, Therefore, I decided to go for a company, and I went to Gujarat to search to hunt for some vacancies in companies. But at some corner of my mind, I had this aspiration of doing science or doing research. And I heard about biotech industrial training program sponsored by Biotech Consortium India Limited, Government of India. I applied to that, and I was selected as a trainee for 6 months through BCIL. Um, I don't think it was a competitive, but because of less applications, I got selected in that. Um, then I went to Bangalore uh, in a company called Symbiosis Biowares for taking the training. The company extended my tenure to 3 more months, like 9 months. I finished the industrial training. The industry was to my fortune, a contract research based institution. So generally, BCIL always gave students opportunity to work in companies, but for my faith, it was a contract research organization, and that was my first step in research. Uh, but of course, it was a limited time opportunity. I came back to Maharashtra and started hunting for job again. But like having a job for 1 year and then suddenly having no job uh, put me under pressure, and then I accepted whatever job was that I got, and that was marketing. But marketing was for a research products from GE Healthcare. I thought it is a good opportunity to touch base with the companies that are doing research in biotechnology. Um, while I was doing marketing, I came in contact with many good people in Spinker Biotech, and one of my mentor over there, he told me if we don't do um, the work of our interest, we can never get satisfied. And I told him I wanted to become a researcher, but because of not having any opportunity, I am doing marketing. He was like, then you are doing injustice with your life. Whatever it takes, do your research. And that way, I started hunting a job in research institutions in Pune, and I got to know about, excuse me, a technician position at National AIDS Research Institute. Uh, as you can see, National AIDS Research Institute is one of the prime research institution from Indian Council of Medical Research, ICMR. Um, but the position over there was technician position, which I thought is like the low division position for me. But my interest in research was so high that I just ignored the designation and started working. And I gave my best to work over there for 2.5 years and learn all the basics of virus, especially human immunodeficiency virus. And I was quite happy to be a part of a very successful project with Central Drug Research Institute at NARI, and also published a research article on that. Then this was a turning point for my life, I will say. In the year 2008, government of India declared that uh, PhD admissions will be carried out through a PhD entrance test. Uh, and I mean, with my master's experience, I was very good at writing the or giving exams. And then I applied for PhD entrance test at my home university, North Maharashtra University. And as expected, uh, as per my expectations, I got um, selected uh, for, I mean, I secured fourth position in the PhD entrance test. And then um, I reached to many um, professors in the institution to see if I get any chance of getting PhD over there. And oops, yeah, so I would like to highlight the contribution of Dr. Satish Patel, who has been my mentor, and he will be my mentor throughout my life. Um, I went to him, and he gave me a small project, uh, which may so he may have to understand if I have the research caliber or not. And for the first time, I tested the drug from his lab called prodigiocin, if it has any antiviral properties, and it had. and I just conveyed him that this drug is fascinating, and it has the antiviral properties. It may have anti-cancer properties as well, because it was inhibiting the cancerous cell that I was using. And looking at those results, he may have uh, this thought that this student can be a good researcher. But of course, I was not like mature enough um, to do science, and he literally um, walked me through the basic steps of doing science. uh, and we had like outstanding association within the lab. We did outstanding research. I mean, the kind of work we did at North Maharashtra University, the rate at which we did the work was fascinating. I haven't been able to replicate that particular productivity till now, uh, though I am in a resourceful environment over here. But that kind of motivation, that kind of inspiration, I am never able to find. So I consider myself very fortunate to be in the environment and under umbrella of Dr. Satish Patel sir, uh, who nourished our scientific caliber um, since the beginning, or from our first step in the research, I will say. Then how did I get the Erasmus Mundus Fellowship, or how did I end up in um, getting my first postdoc position at France? That's the question. We don't know how to actually apply to the foreign institutions for, especially for postdoc or a PhD, whatever it may be. But the Herculean task was actually done by my PI, my supervisor, Dr. Satish Patel sir. He, I remember those days when he used to write at least 10 to 15 emails to different researchers across the world to see if he can get we can get the opportunity to do research over there, because when we are at the PhD level, uh, we had a limited scope of um, getting answers from big researchers. But if it is a PI who is writing for you to the big scientist, then there are chances that they may get reply. And um, uh, his philosophy of applying to different or emailing to different scientists was that what can happen, what can go wrong if we are writing to him, if we are requesting to him to get collaboration with us, to do collaboration with us, or to give opportunity to one of our student. What can happen? He will say no, that doesn't hurt us, we can keep on writing to many people. And he did that Herculean task, and got contacts all over the world, not only for the future opportunities for the students, but he also got uh, us to get in collaboration with researchers across the world. I know for my PhD, he approached uh, scientists from France, from USA, and from Brazil, where my work was carried out. And that's why it has been so impactful. We published almost 30 publications in the 4 year time, which is fascinating, with 6 first author papers, and that increased my merit to be eligible for the outstanding fellowships like Erasmus Mundus that I applied for. So one more interesting story about Erasmus Mundus Fellowship is, um, when I applied to this particular position, I was advised to apply it by my senior Chandrasekhar Patil, but he didn't tell me that there are only 2 positions all over the country. I mean, only 2 candidates will get selected for the postdoctoral fellowship. Uh, and I just applied it from my end. And then when I got selected, I got to know that there were only 2 positions, and I am one of the only 2 candidates selected for the fellowship. This was like so inspiring, because if I would know that there are only 2 positions for this postdoctoral fellowship, I would have never applied, because that would have created a negative mindset in me that, oh, there are only 2 positions and we are like 100 crore population, out of which at least 1 lakh students are uh, have the same skills that I have. Then why should I apply? It will be a waste of time. But it is never a waste of time. uh, I'm very fortunate to not know that there are only 2 positions for this particular fellowship. But then my journey to the foreign countries started, and of course, I never looked back from then. Uh, I finished 10 months of my postdoc in France. I worked on metabolomic approaches, which was interdisciplinary for me, but I still took it like sportily, and I learned the chemistry, though I didn't had the chemistry background, but that actually um, built a adaptive ability in myself, I will say, to uh, accompany people or researchers from interdisciplinary sciences, and actually gave me a different direction of research thinking. After 10 months, of course, uh, what should I do? I mean, I never knew what should be the next step. But again, my mentor told me, stepping down is always easy, but um, if we just think only one step ahead, then climbing is not impossible. And keeping that in mind, I started to apply for more fellowships, and I got more failures. As you can see here, I applied for a fellowship from Singapore, I applied for a fellowship from Israel, uh, a fellowship from Switzerland, a fellowship from USA, uh, but I got failure. Then I thought there is something that I am doing wrong, and I was I decided that until I am in the France, until then only I need to find some position within those last 4 months that were remaining by then. Then I searched for different fellowships. I tried to establish a contact with different scientists. I worked the same way that my teacher taught me how to do that. Then I read the science of the researchers and try to come up with some research proposal which can be of mutual interest, and then contacted researchers across the world. Um, and I was able to get connected to a number of scientists, but was not able to get any fellowship as such. But then, after trying so hard for a while, I was very fortunate um, to get into the lab uh, with Dr. Shukla at US, and I worked for 3 years over there. Uh, and now my science begins. So here, I would like to switch on to the science, how the science actually motivates me. Until now, you saw how my different steps in my career motivated me to climb up. But science is my real motivation, and I will talk briefly about it. So I started after working on HIV back in National AIDS Research Institute. This was the second time when I was working on viruses, and the virus I worked on was a herpes simplex virus. You all may be familiar with the herpes infections that are very common among people, and I think herpes simplex infections are such prevalent that more than 70 to 80% population all over the world harbors it. The only time the infection appears is when your immunity is down, when you have fever, or when you have um, recovered from a major disease. Then you can see some scars on your face, or here in US it is pretty common to have infections in the ocular site. Um, while studying with the virus, I first underwent through the literature that is available on herpes simplex virus, and I did see that the virus first infects the corneal surface, and then through the path of optical nerve, it goes to the trigeminal ganglia and then to the brain. So it doesn't only cause ophthalmic infections, but it also causes encephalitis, which is infection to the brain. And in rare cases, this encephalitis causes death of the patient. But you know what? Though more than 70% population is infected with herpes, only rare events are there when the death of the infected person occurs. Uh, so this particular question triggered me to decide my research topic is what may be the reason how our body may be protecting itself from the severity of the disease, or in the other words, um, how our brain deals with the virus infection and saves our life. So while studying that, I closely observed the virus life cycle. Our body has so virus, it first enters inside, it replicates in the nucleus, then it packages its proteins in the Golgi apparatus, and then it matures and bursts out of the cell. So this is the like normal procedure for the virus replication. But what happens in the body, like at which stages the body actually controls the virus replication, that is more important. And here we can see that body can either stop the entry of virus, stop the replication of the virus, stop the packaging of the virus, or it can produce the antibodies against the virus to neutralize the virus before it enters in the cell. But one other mode of infection inhibition is in death of the infected cell. This we can call it as a suicide of the cell which is infected by the virus, because when the cell death occurs, the pH of the cell increases, and that also neutralizes the virus. I mean, it degrades the virus within its side so that it cannot spread to the other cell types. But you know what? If this cell death happens in the brain, then brain cells are very weak to renovate for the renewal, and the brain cells can go dead, and that may actually cause paralysis or even death of the patient. And understanding that, I first wanted to know if the virus infection is actually causing the death of the individual. And for that purpose, I did a small experiment with a live cell imaging. Uh, may I ask if you can see the slide? One second. I'm actually
No, sir,
playing a
it's not
video.
visible.
One second. Is it visible now?
Yes.
Okay. So this is very interesting. The power of technology we can see here with the live cell imaging system. Actually, we can see that. So these are the cells, and the red which is appearing is the virus. And we can see that the virus grows in the cell, and then sometimes the cell die. But of one more thing I will show you. See, this is a different cell type. It has a mutation for one of the host factor, and you can see such a huge virus replication is occurring everywhere. As you can see, the virus is spreading from cell to cell. Uh, the virus spreads from cell to cell and causes more replication. But sometimes when the cell has or the when cell lacks the properties, certain properties, we can see the cell death happening that limits the virus spread. You can see all the infected cells are just dying and they are not spreading the virus infection to the neighboring cells. This is one of the strategy employed by the virus, by the body, to control the infection or to limit the virus spread. Um, I wanted to see the similar effect in the animal cells if the cell death is common mechanism in them as well. So I infected mouse with the virus, and then I took cross sections of the eye of the mouse and brain of the mouse. And as you can see here, um, if you can see the red spots along the line of the cornea, these are the cells that are dead, and that happens because of the virus. Similar, in case of brain, we can see some red spots, which are indicator of the death of the cell. So this was clear to me that cell death mechanism is active in in the organs infected with the virus, and therefore I decided to go ahead and work on the factors that are actually causing the cell death. Here, I went through the literature and I came across a factor called mTORC2, a mammalian target of rapamycin complex 2. This particular factor had the ability to activate the immune system and also to activate the cell survival. And these both processes are very important in during virus infection, because immune system controls the virus replication by creating antibodies, whereas cell survival survives the cells by inhibiting the stress situations or stress signaling that is activated inside the cell. With this review of literature, I started working on mTORC2. While working on mTORC2, there was one mm, major limitation, because there was no inhibitor to work on this. Therefore, I took a component of mTORC2, that is Richter. Richter is an essential protein without which mTORC2 is not functional. Then I checked if Richter expression, protein expression of the Richter, is it different inside the cell during virus replication. So this I'm showing is a Western blot, and the black lines that you see is actually a protein, and HPI is hours post infection. So 1, 2, 3, 4, 5 till 12 hours post infection is the time period. And I did see when the virus starts replicating its protein, the expression of Richter either decreases or increases. It gives me an impression that daya kuch to gadbad hai. So Richter has something to do with the infection, and I decided to go ahead with working on this particular molecule. And as I said before, it was difficult to work with this host factor or host protein because there was no model available for working. Then I made a genetically modified mice which lack the expression of Richter. So this was again a challenging job. It took me at least 6 to 8 months to breed these animals and to make them genetically modify for Richter. But when I use these mice, I will call them as a Richter knockout mice. Uh, these Richter knockout mice were infected with the virus, and surprising results were obtained. This is a survival curve where the green line represents the death of the animals, whereas the green line is actually a knockout line, and pink is or purple is the wild type animals. Wild type means they do have a Richter, they do not have knockout. Um, and when I infected these knockout versus wild type animals, I saw 80% animals were dead. That means Richter or mTOR-C2 has important role in saving the life of the infected animal. I took account of the virus production, and the virus production was also more in knockout. That means presence of Richter was actually able to control the replication. And you can see here, after infection, ocular infection of the mouse, there were phenotypic differences between the knockout and wild type. So knockout looks more infected than wild type. Then I wanted to see if cell death is different in both these knockout versus wild type. I forgot to symbol these eyeballs, but this is a wild type and this is a knockout. As you can see here, there is a limited amount of cell death in the mice having intact mTOR-C2. So when mTOR-C2 is there, there is less cell death. And when I infect them in the cornea, this is the eyeball, you can see all the lining of the cornea shows cell death. And the cell death is actually along the neuronal line. So this is the optical nerve, and it is going towards the brain here. And it was so interesting that motivated me to see if we do see the same effect in the brain as well. And it was right. I'm sorry. In the brain sections also, I did see the cell death in case of knockouts compared to the wild type. This motivated me uh, to hypothesize that cell death during infection is controlled by mTOR-C2 or Richter. But again, what was the mechanism behind the cell death of Richter or mTOR-C2? Then again, I went to the literature and read the work done by others and tried to connect the dots. So nobody explained how actually mTOR is linked to other survival pathway, AKT, and then FOXO3A. So here I'm using some technical terms called AKT is also a protein, cellular protein, which have role in the cell survival. And FOXO3A is a transcription factor, which has a role in apoptosis or cell death. So because these all three proteins sharing same role, I thought they may be connected to each other. There may be a signaling that crosses that actually transduces the cell death happening during virus infection. So after this hypothesis, I studied the location of FOXO3A in the cell. And to my surprise, this was like most exciting moment of my life, I will say. I remember it was 11 at night. I was sitting on confocal microscope and was watching the cells, uh, and I labeled the proteins with red and green and blue. And I saw, particularly in virus infected cells, the location of FOXO3A transcription factor was out of the nucleus. So to explain this properly, the blue is the nucleus, uh, and the red is the FOXO3A protein. This is what I postulated. FOXO3A, if it is inside the nucleus, it causes cell death or apoptosis. But when a virus infects a cell, it pushes the FOXO3A protein outside the nucleus so that it doesn't have any access to the DNA, and the cells doesn't go uh, towards death, and it uses the machinery of the cell to propagate itself. So this was so amazing moment. It gave me goosebumps, uh, and this fact was almost opposite in FOXO3A knockout animal. Sorry, in cells. I did see the FOXO3A presence inside the nucleus, and there was very low amount of virus replication happening in these cells, because if the virus infects a cell, it will immediately die. So it will not let the virus use its assembly. That way it can protect the neighboring cells from getting infected. So this was so exciting, and that night I wrote a statement on my desk that I heard the rhythm of the life which was beating since the genesis of life, and I was the first one to listen to it. This was a very amazing experience for me. Um, but then I also checked the same mechanism in the neuronal cells. Previously, I checked in the corneal cells, then I checked in the neuronal cells, and it was the same mechanism of cell survival. And then um, I use some inhibitors to inhibit the cell death, which actually protected the dying animals. So if you treat the infected animal after certain days with the cell death inhibitor or apoptosis inhibitor, you can actually survive few animals from the group, which was exciting. That means my research can actually save the life of infected people. Uh, this was so exciting to know. Um, then I studied other aspects of mTOR, which I will just touch briefly. mTORC2 was found to increase the immunity of the body, and as we can see here, this is also the cornea. And in wild type animals, we do see a lot of immune infiltration, immune cells, but in the knockout infected, we do not see much immune infiltration. Then I studied the cytokines. I know you guys are familiar with these terms now, because with COVID-19 we do see that in patients there is a cytokine storm. So so those cytokine storm like situations are in the wild type animal, but that actually contribute to decrease the viral load in the animal compared to the knockout, where we do not see much cytokine storm. um, so it creates a defective immune response if mTORC2 is not active in the body. It also causes less production of the neutralizing antibodies in the knockout compared to the wild type, and this was so exciting. And I'm proud to say that this finding was published in one of the very esteemed journal called Nature Communications, and I'm very fortunate to be part of it. Of course, I'm the author of it. Then I approached to the next step. When I actually approached to the next step, it was a COVID pandemic that was already started. And being a researcher, I thought it is more important to apply my skill set to the SARS-CoV-2, which can save the millions of life. And then I applied to a position at Gladstone Institute. By then, I had a strong portfolio of publication. I had a very good skill set, and of course, it contributed to my confidence and to join the Gladstone institution. This was the only application that I did in last 3 years, and I got selected for that. That shows what kind of caliber I might have gained through the research in past few years. Here, my responsibility is to establish an animal research facility. Of course, it is a bigger responsibility to execute animal research for collaborators and do my own research and write grants and whatnot. But since was 1 year, I'm working very hard to execute my responsibilities properly, helping everyone in their research. That increases my interaction with the people, and I gain more and more knowledge. So this is also one thing that I want to highlight. If you are being more helpful to others, it contributes to your overall development. And many times people actually help you uh, uh, without knowledge of yourself, and that actually benefits you to progress further. So here I am basically touching base with the entry mechanism of delta I am currently working on, and started simultaneously working on Omicron. As you can see, delta is the biggest or the highest runner, was the highest runner in the race, but I think it is Omicron now. And we are a micromolecular virology lab where we modify the virus uh, by inserting or deleting the mutations to understand what is the role of a particular mutation. Uh, um, and as I can see, the time is running very fast. Just a couple of more slides, and then I will wrap up my presentation. So being a scientist is, I feel very special about it. Discovering secrets of life, of course, bring me closer to the mother nature. And it gives me a sense that I have the ability to save millions of lives with my science. And of course, our publications are there forever. So with your publications, you will be remembered forever. This is the only job where people pay you for you to get famous. So with that, I will say life is a relay race where our parents work hard for us. They hand over a flag to us, and then we start running further to give that flag to the future generations. And I will also say that I have been a marathon runner, not a sprint runner. I mean, not technically marathon runner, but I want to tell you that live your life, or see your career as a marathon and not a sprint. You have to run continuously, work at full pace, but also save energy for your persistent ability to work continuously, and not like put all your energy in one thing and just end up in getting tired and then just have rest. That is not how you will get the success from your college life. So with that, I will say grit is also one of the quality that I think I had, which motivated me to come along the way, and to contribute myself or my knowledge towards science. Uh, with this, I would like to acknowledge my parents, of course, for everything they have done for all their life. Uh, there are no words to say thanks to my mom and dad. Then School of Life Sciences, as I mentioned before, my mentor, Dr. Satish Patel sir. Uh, I'm here because of his contribution. He did every possible thing to let us reach here, and I always will be grateful to him. My lab members, uh, my seniors, everyone have contributed a lot to my scientific advancement. My postdoc mentors, Dr. Deepak Shukla, and current supervisor, Dr. Melanie Ott, and special thanks to um, Dr. Anand Mohan Chakravarti, uh, to whom we lost last year in Chicago. He was a great inspiration for me, and will be a great inspiration for me for all of my life. Um, and of course, I cannot forget my friend, Little Mouse, who sacrificed his life for all the humans to save their life. Uh, with this, I will thank you, everyone, and I'm open to answer any question if you had. Thank you.
Thank you, Dr. Rahul, for the very inspiring and motivating talk. Dr. Rahul has nicely explained that how, we have any idea, so how we can put it into reality with our hard work and passion. He nicely gave that what actually the current status we have for the SARS or COVID virus, and what is this overview, what the experiment he has done to search it out, what is the mechanism, and what actually been the possibility for the treatment or for the drug. He nicely give his overview regarding its path, that from where he has started, how he said that he was a very average student, but from that average student, how he progresses, what are the obstacles he faced, and how he passed it. Actually, this has been, we can say, like a story of every common student, and really Rahul has given overview of his research, and from starting from his education, what are the obstacles, what are the actual things he found or faced, and how actually he tried to get come up from that. So thank you so much, Rahul. It's a very really a motivating for all the students. So after listening such an interesting session, because it's been a motivating session, but it's having the audience from various faculty of sciences, including the other than the biological sciences also. So he restricted his some of his research to a limit, but maybe the biology questions or the other, all the other students also may have some of the questions in their mind. And so now we move on with the other further session regarding the question and answer part of the session. So I request Dr. Rahul to please respond to the questions from the participants. So we have a number of questions that the students had put in the chat box. So for this question and answer session, I call upon Dr. Ulka Malude Madam for the question and answer session. Over to you, Madam.
Very good afternoon to one and all present here. And thank you very much, Dr. Rahul, for giving a very wonderful talk on a motivational innovator, a successful innovator. I think the students are much impressed by your talk and the hard journey which you have during your life. So, the questions are from student. Here I want to mention, the question is from Aishwarya Charode. She is a MSc student. How does Richter work during self-death? Her
Yes.
question.
Yeah, so as I showed the schematic, Richter actually activates one of the protein called AKT, also called as a um, protein B, protein kinase B. And then AKT goes to the nucleus and binds to another protein called or phosphorylates another protein called FOXO3A. So basic function of FOXO3A is to cause cell death. But if AKT binds to the FOXO3A, then FOXO3A conformation of the protein changes and it can no longer bind to the DNA. If it cannot bind to the DNA, then it has no right to be in the nucleus. So FOXO3A is exported out of the nucleus. So when FOXO3A is exported out of the nucleus, the cell death pathway stops, and that way the cell death mechanism is hindered. If this mechanism is operative in the neuronal cells, then it actually saves the neurons from degeneration and it ultimately saves the life of a person or patient.
Okay. The next question is from Vaibhav Manmode. He want to know certain information from you that he is interested in industrial job, not in research. So what can I do for industrial job?
So even in industry, I think the I don't know if he's from the biology background or chemistry background or
He's
any other
from
background.
microbiology. He is from
He is
microbiology, MSc
okay.
microbiology student.
Then in that case, I think the pharmaceutical companies or the research oriented companies are very welcome to postgraduate students or even the researchers for working in their companies, like company Serum or Bharat Biotech, or from the big pharmaceutical companies like Pfizer. They have the openings, but to my knowledge, the only problem with the companies is they do not publish their vacancies on the public portfolios. So you need to be in touch with your seniors who are already working in the industries, have their references. By that route, you can get a chance to get interviewed, and if you are knowledgeable enough to benefit the company, they will for sure hire you. So major portion of the students, or more number of students, actually prefer to go to the company uh, because it is a quick source of getting a job.
The next question is from Aishwarya Gautam. She wants to know, is COVID-19 variant Omicron worse than Delta variant?
So there are two things. One is it has higher entry compared, or higher co-transmission, I will say, compared to the Delta. That is for sure. But looking at the severity of infection, I think the infection is on the milder side. So Omicron patients may not face like severe situations because uh, because of vaccination, people have already acquired herd immunity, and it is although Omicron is infecting people, despite of having robust antibody response, people are still getting infected. Vaccinated people are getting infected, but I think the probability of getting to severe disease is less, and this is a good thing about the adaptation of the virus. And um, but of course there are some immunocompromised people, old people, which would not like to have the infection, though they have antibodies made from the vaccination. uh, the Omicron may contribute to increased severity in them. Currently, the studies are ongoing of to know how effective different vaccines are on the Omicron, and I think booster doses of um, vaccines like Pfizer and Moderna are enough to get more immunity against Omicron that has been revealed in past since past week. But the studies are ongoing, and we will have more answers in next couple of weeks.
Sir, one more question relevant to Omicron from Shivani Kaude.
Yes.
Which type of mutation occurs in Omicron variant?
Major mutations in Omicron are happening in the spike area. So uh, when we compare Delta, which is like most pathogenic variant till now, in comparison to Delta, Delta has like 11 mutations in the spike area. Spike is a protein where which a virus uses to attach to the cell, and this is the same protein against which different vaccines have been designed against the virus. So if there are more mutations in the region, there is a less possibility for a vaccine to be effective. And in Omicron, we do see more than 30 mutations in the spike area. It also have mutations in the envelope region of the virus, which contributes to complexity of neutralizing the virus in infected people. So that way it is more dangerous.
The next question is from one of the MSc student of microbiology, Nikita Moe. Her question is that what to do in gap between MSc and research?
Sorry, can you repeat?
What to do means after completion of their PG, MSc, and
Okay.
before joining the research institute or any research, the, what
What
should
should we
be
do?
done by them?
Okay. So the first thing, if you want, if you are interested in research, I think nowadays you need to get PhD, because it is a basic qualification required to do research. It actually teaches you how to do science, how to do research. So I think you either need to get qualified, different competitive exams like NET or PhD entrance test from different universities, and then you need to get enrolled under a professor which who is ready to guide you for your studies. And once you complete your PhD, then actually you can get more chances to work in science. And as I explained, you can apply to different fellowships available that can give you more opportunities to work on higher end research.
Okay, the next question from Jayashree Bahe, that is Omicron is less lethal?
Um, so I should not comment on that, but until now, like my own observation uh, of reading literature about the Omicron in the news articles, basically, we shouldn't believe the news articles a lot. We need to wait for the research articles to get published, and we can comment. But the hospitalization studies on Omicron patients are not so severe. Uh, the the other reason for this is also because um, generally the youth population who is not wearing mask in South Africa or in the other countries is actually more prone to have infection. But since the beginning of pandemic, we do see that to the youngsters the infection is less severe. So it may be the reason, because it is getting spread in the young population. It is, we are not seeing the severe cases. But uh, if it is infecting to the older population, then only we can comment on the severity of Omicron compared to Delta or other variants. So I think we need to wait for a few more weeks to get the answer for this.
The question is from Aniket Thute. Why do all researchers choose to prefer experimentation on mouse other than any other animal?
So mouse is a great tool for research because we can get many mice in short period of time. It has a gestation period of only 21, I think 21 days. Within that period, you can get like uh, 8 to 10 animals from one breeding pair. That is great. Um, the other thing is we know the genome of the animal, and we can actually mutate the animal for different host proteins that we want to study for infection. So not only for virus infection studies, but Alzheimer, for cancer research, for any other disease type, mouse is a very good model. But to just answer this question, uh, mouse is not like the best model for doing SARS-CoV-2 research, because SARS-CoV-2 specifically infects humans, and we are using genetically modified mice where we express the receptor of human, that is human ACE2, in the animal to make it a suitable model for virus infection. But um, the genetic diversity that is observed in humans is not replicated in mouse. Therefore, I am working on another project where I am using genetically modified uh, diversity outbreed animal. These diversity and outbreed animals are a combination of 7 different mouse populations uh, available throughout the world, and they have been crossbreed with each other to get the genetic complexity. And we are going to test the virus infection into that, and we expect to see uh, the or recapitulate the human phenotype that we normally observe in patients. So we are hopeful for that, for good results from that study.
Okay, there are so many questions in chat box. Are you comfortable with this question?
Yeah, I
Shall I
am
proceed?
comfortable. If your time permits, I am okay with that.
Okay, okay. One more question from Pratiksha Deshmukh. She want to know, where did Omicron get its name?
Okay, so this naming is happening according to the letters alpha, beta, gamma, delta, lambda, epsilon, like that. So Omicron is the number from the series itself. Before that, there was a psi and one more number I don't know, but I think because of the controversy, because uh, many Chinese people have the name psi, so it was not relevant to give the same name to the virus. Therefore, they jumped to Omicron directly in order to nominate this or label this variant as a Omicron. So it is just a series of the number, and they are giving the name. I mean, WHO is giving the names of the variant according to that series.
The next question from Kshitij Bhavsar. In considering Covaxin, does it be more susceptible to the virus?
I don't think I have read any data on Covaxin and its ability to neutralize the virus. Uh, every vaccine company is actually doing study to know the efficacy of their vaccine against the Omicron. Like we are studying for Regeneron and few other companies to know if their vaccine is efficacious. But to my knowledge, I don't have any idea of Covaxin, how it is effective or at what extent it is effective. So I think we have to wait for their data to publish.
Okay. Isha Deshmukh wants to know, what is the most challenging part of being a researcher, and how you keep yourself motivated to face those challenges?
As I explained a few times in during my presentation, you get passionate to know uh, the um, the to listen to the rhythm of the cell, and you, if you are the first one to listen it, then it motivates you. For me, the motivation is whenever I see under microscope, I did see amazing things inside the cell, inside the virus infected cell. I label different proteins, I check their location, I check how they behave, and any new thing actually excites me more to dig more deeply into the subject matter, and and learn more about the actual science behind it. I think that knowledge actually helps uh, to modify or to bring up a new treatment modality against certain diseases, and that way I actually find my motivation. It's on daily basis actually. Whenever I work with the animals also, uh, we just not harvest the organs, but we closely observe different um, systems in like digestive system, reproductive system, respiratory system, nervous system. All these systems I observe very carefully in each animal to see if I'm observing any significant phenotype that may contribute to the advancement of science. So I find my inspiration, my motivation by myself. Uh, Anybody who wants to work in research, I'm very sure if they are working passionately, they will find their motivation in their day-to-day work. Yeah, that's what I will say.
Madam, we are short with time. So you can take one last question from Dr. Sunil Koi, and then we can stop the or finish this question and answer session. Please.
I request all, may I interrupt you, ma'am? I request all the participants in between, please start your video. so that we
Yes,
can have a photograph
after, yes,
afterwards.
yes. After this session, question answer session, we actually going to take the snapshot.
Okay, the question is from Dr. Sunil Kohli. He wants to know what is your opinion on the co-infection of bacterial pathogen in COVID patient?
Um, so it is a unstudied question till now. Uh, I will say not only bacteria, but any other viruses or any other microorganism, when there is a co-infection that occurs, it needs to get studied deeply, because sometimes it may happen that some other microbe infection can have opposite effect on the Omicron phenotype, on the SARS-CoV-2 phenotype that we see with these different variants. For example, it has been seen that if you have immunity against a certain um, pathogen like TB, um, then there is a chance that you get cross-reactive antibody response against the SARS-CoV-2. I mean, I'm not like very particular about TB, but I'm sure people are doing different studies on co-infections and checking if they have a cross-reactive immune tolerance, or if it is matching with the current infections in people. There are also studies in autoimmune diseases. In autoimmune diseases, we have constant immune response. I think which can increase the innate immunity to a level that when the virus actually enters in your body, it will get neutralized sooner compared to those patients who are not having the autoimmune disorders. So it's not only about the co-infections, but even other diseases might have the impact on infections of SARS-CoV-2 or even other viruses. But it is a very important area of study, and I'm sure researchers are on it.
Okay. Thank you, Dr. Rahul, for a good interaction with the students. And from your, and I'm very thankful to you that you share your story. So from your story, the student must understand that nothing is easy, hard work is required, but nothing is impossible if your determination is strong. So
Yes.
hope the student understand how patient we should have during research, and how to tackle the problem which come across in our life. I again thank you. Over to Bhavna, madam.
Thank you so much, ma'am, for nicely conducting this question and answer session. And thank you to Dr. Rahul Suryavanshi for his enlightening talk and solving the queries raised by the participants. So thank you so much, sir. I request all the participants to switch on their video so we can take a group snapshot. All are requested to switch on your video, please. All of you, please switch on your video. Please keep your video on for some more minutes. Yes, thank you all of you. You can switch off your video. This is an instruction to all the participants. The feedback link has been given in the chat box. So you can fill the feedback form through this link and you will receive the e-certificate after filling that feedback form only. So please take a note down of it and fill the feedback form. Certainly within a week, you will get your e-certificates on your email ID. So now we reach to the end of this event, the motivational talk by a successful innovator, Dr. Rahul Suryavanshi. So I call upon Mr. Mayur Hite from the Department of Microbiology for the formal vote of thanks. Over to you, sir.
Am I audible, madam?
Yes, sir.
Good afternoon, everyone. Once a great man whispered, feeling thankfulness and not expressing it is like wrapping a present and not giving it. So, on behalf of Department of Microbiology and Institutions Innovation Council, I take this opportunity to put all my gratitude into words. I would like to thank our principal, Dr. Om Mahoday sir, who graced us with his presence and guidance. Thank you, sir, for giving permission to organize this program and sharing with us your words of wisdom. I am delighted to thank our guest speaker, Dr. Rahul Suryavanshi, for enlightening us with his knowledge on innovation. Today's program was full of knowledge and motivation, and Dr. Rahul Suryavanshi make it very interesting. I also want to thank Dr. S.S. Khandare, convener of today's program, and head of the department, President of IIC, Dr. N.P. Mohabansi, convener of IIC, Dr. B.B. Moite, coordinator of today's program, and all the faculty members for organizing this event and making it successful. Last but not least, I thank all the students and participants for showing their interest in the program. I once again thanks everyone for making this program successful. Thank you very much.
Thank you so much, sir. So with the permission of the chair, I conclude that the program is over. I am again thankful to all the dignitaries and participants for their attendance and active participation. We hope this event will be of value and it will certainly give some direction for your further future ideas. Thank you so much to all of you. Stay safe and healthy. Goodbye to all.